Can you adjust your camera angle at all? Or yeah, you were very much like a lot of
Hi.
yeah.
I'm Michael Nachman. I'm a professor of integrative biology and the director of the Museum of Vertebrate Zoology at UC Berkeley. We'll explore the evolution of rock pocket mice.
Dr. Bohr, I'm so excited to talk with you today.
And we then also got very much into mitochondrial function. And so in my work, I've tried to combine studies of the mechanism of mitochondrial function and DNA repair. Ended up with work that relates to neurodegeneration.
You are colorblind, or should we say color vision deficient? Do you have a preference there?
I don't think about it that much, but yeah, I wouldn't call myself colorblind, but I'm color deficient. Yeah, it shows up mostly on my poor choice in clothing.
I'm speaking today with Dr. John Molt.
So being able to say, what a lousy idea, but what does that tell us about what might be a better idea? What might be a better algorithm for trying to tackle this problem?
We have this animal that we think is a red wolf, but we need you to think it is too, right?
Yeah. So what's really wonderful is we're gathering lots of data on the ecology and how animals that have more red wolf DNA, they are acting quite different from a coyote, something that has very little red wolf DNA in it.
If we're able to show that, lo and behold, the red wolf, through its sort of top carnivore role, was actually, and that maybe by returning red wolves to other parts of the Southeast, we could actually boost this really popular game bird, the quail. I think that it has the potential to be sort of a game changer for how land managers look at red wolf restoration.
I'm Ralph Peterson.
And I'm Sarah Hoy.
And we're both researchers at Michigan Tech University studying the ecology of wild boar.
We're looking forward to sharing our research with you.
It's interesting. I was working with a teacher. She had talked about giving the 23andMe. She'd done testing, and then it came back, and it's like, here's your 2 brothers and like 2 sisters, and she's like, I only have 1 sister, and then it was like,
oh wow.
So we're going to go ahead and jump into this. So let me go ahead and just again welcome everybody. I'm Noel Pauller. I've got an intro slide coming up here, but so we're talking tonight about teaching AP biology with storylines, and I'm sharing with you some more information about the storylines that I developed for AP biology. So you get some of the backstory, some of the origin story of the Lab Hamster storylines, and there are a number of questions that got posted to the Facebook group as I, requested questions to come in. What are people curious about? What are people wondering about? So my goal is to try to address as many of those questions tonight as we can. Okay, let me check here and see. Okay, all right. So again, I'm Noel, and so I'm an AP biology teacher. I also teach standard biology. I teach a STEM science class. I've taught chemistry, microbiology, earth science, just a bunch of things over the years. So my degree is a master's in education and life science education. I celebrated last December. I got my national board certification completed, which is, if anybody has done that, or if anybody is looking into it, it was definitely something that taught me a ton. I really had a great chance to reflect. It's a lot of work. I currently teach at North County High School, which is in Glen Burnie, Maryland. If you're not familiar with Maryland, we're just south of Baltimore. North County is part of the Anne Arundel County Public Schools, where Annapolis is located. So we're sort of between Baltimore and DC. I'm the writer of the Lab Hamster storylines for AP Biology. I'll give you the short version of, thank you, Matthew. So I'm going to give you the short version of the origin story of the storylines that I wrote. So back in November of 2023, along with a coworker, I attended the NABT conference when it was held in Baltimore. It was basically just right across the street from my school, and attended some sessions on storylining, and thought that this is the direction I really wanted to go with my teaching. So I had been teaching at that point AP Biology for 2 years prior to that. And I first started out where the first year that I taught it, oh, and I should tell you, so this is right now, this is my 25th year teaching. And for the bulk of my career, I wasn't teaching AP Biology. In fact, there were years that I kind of had turned it down earlier in my career for a number of reasons. And at that point in, like right after COVID, my department chair came to me and said, I really want someone else to teach AP Biology with me so that, you know, that I have someone to collaborate with and we can make the class, you know, stronger class for all of our students. And so we were really seeing a big problem with a lot of our students where there were a lot of students who weren't really engaging in class. You know, they would show up, right? And they'd registered for the class. And I think that, you know, on some level, they wanted to learn biology at a high level, but what we were doing wasn't really connecting. And when I went to the storylining presentation, I thought, this is maybe the way to, you know, reach out to some of these students who are deciding they want to take the class, and then they're in the class and they're just not, for whatever reason, they're not really seeming like they're enjoying the experience, right? Or that they're invested in the experience. So instead of just saying, well, this is the student's issue, you know, Angela and I, you know, took a look at that and said, well, how do we make this class better, you know, engaging for students? Hello, Joe. So, you know, when I heard the storyline presentation, I just thought this is the direction that I want to go. So then it was, well, what storylines do I use, right? And there weren't any for AP Biology. And in hearing that presentation, I kind of discovered that where I was moving, the direction that I was going, was already really close to a storyline. In some cases, the changes that I needed to make to turn the existing unit into a storyline unit were not that big. Now there were some units where it was further away, and there were some units that were trickier to come up with storylines. And I'm still learning as I go. I'm still trying to modify these units as I teach them and to reflect on what's working and what's not, and to constantly make them better. But so I started that fall. I think Unit 4 kind of did a storyline. It wasn't really, if I look back on the format of what that first one was now, I'd probably say like, ooh, I was, you know, learning. But then kind of picked up on how to do it, did better with Unit 5, and then 6, 7, 8 that year I felt were pretty good. I was working on my NBCT. So by the time I got to Unit 8, I was really like invested and really kind of hitting my stride, and then time to go back and you know, be able to make some changes to the prior units. And then so by the end of that 23-24 school year, you know, felt like, hey, I really got a handle on this, and I can go back and, you know, really edit all the existing units and get them to where I think they're pretty good. Yeah, so that's kind of the origin story. Why is it called Lab Hamster? That's a whole separate story. That would take a long time. So we'll do that at another point in the future. Okay. So, you know, storylines, you know, many of you are here because you're interested, but maybe you're on the fence. Maybe you're thinking, this might be a fit for me. It might be a good thing for my students. Maybe it's not the right thing for me and my students. So why teach with storylines? Well, for starters, I think they're more fun, and I think that they're more interesting. I'm enjoying myself doing my job teaching AP Biology more now than I did before. And I liked my job before, but it's better now. You know, so that's one piece. And I think that the students are enjoying the class more too. You know, I think that this really stems from that these storylines, I think, are really engaging. I think they're really gonna capture students' interest and get them invested in the learning process. In addition, they really spark, students being inquisitive. I think that the storylines can really move students from a place of feeling like this is the class where there's a bunch of information that I have to learn to this is the class where we ask a whole bunch of questions that I would like to try to find answers to. And I think that that's a really important shift to really kind of encourage that curiosity in students. Storylines are using inquiry to help students answer their questions. Now it's not loosey-goosey, it's very guided, it's very structured, and there's a lot of interactions, between teachers and students. So they're not just, I think those early models of inquiry, for those of you that have been around, there were early models, I think, of inquiry that gave students a lot of freedom, and in some cases too much, so that they were kind of lost in the sauce with the process. And I think that another thing that I've seen is that they're raising my students' achievement. I'm seeing students earning better grades. I'm seeing students completing their work. I'm seeing students do better on unit exams. And we saw a really nice jump in our school AP exam scores last year with a half of a year implementation. Now it's a fairly small sample size. It's 1 year. So, I'm not going to say I know for certain that I've got statistically significant data that says that these storylines are going to, improve every student's performance. But I think the level of engagement, that students are coming to class and I don't see students checking out, they're engaged every day in class. And I think that that's a big key to students performing well in the class and enjoying the class too. Okay, so there were a number of questions that got submitted, and I wanted to just share a bunch of these, right? There were a lot of them that came in. So how do we make sure that we're covering all of the science practices, all of the topics, all the course content that's included in the CED? Oh, I got this here. Again, another person was asking, how do we make it through the whole curriculum? Some teachers shared they start after Labor Day, and we still have the same exam date for everybody. So how do we make sure that we're getting through that entire curriculum? How do we organize and use textbook resources and AP classroom questions? For example, those personal progress checks. How do we use those effectively in a storylining model for AP biology? Is there a way to minimize homework? How do we encourage students who might not be motivated to complete homework? One teacher who posted this question shared, that she has a high number of students that may not have some of the resources to, do homework. And how do we encourage students to make sure that they're following through on that end of their learning? How do we use storylines, and deal with student absences and makeup work? And then what kind of supplies do the students need to stay organized? And so this is kind of a summary of the questions. Now, before we really go a ton further, are there any other questions that some of you had that you feel are different than ones that are here? If you do have one that you feel like I haven't quite captured it in the ones that came in through Facebook, what are some of the other things that we would like to that you all would like to talk about or find out about? How does the use of AI impact the use of storylines? That's an interesting one. There's actually a unit incorporating AI, but I think you're meaning student use of AI, correct? Yeah, let me make a note of that and kind of think about that. And we can, what's the benefit of the digital notebook versus paper? We're going to get into that. Students keep a paper notebook. So yeah, but we'll talk about notebooks. There is a digital notebook. Mostly student use, of AI. Yeah, I mean, I think that it's out there, right? Is there a higher cost of lab consumables? That one wasn't asked before, but that's a great question. So let me put that down too. And operating on a $0 budget. Okay. All right. So I think I've got good news for many of those things. That sounds hard to go on a zero budget. Yeah, Title I school. So my school is not officially Title I. I have a feeling that if our data tracking was fully accurate as far as SES data and family income levels, that we probably actually are a Title I school. But officially, I think some of our families are not reporting accurately, so that we're just below cutoff. So I can certainly relate to teaching in a school that's a Title I school or one that's close to it. Now I've also been on the flip side where I've been fortunate in my career to teach in a really wide variety of schools. So I've been at very affluent communities. I've been very kind of middle class, and then I've been in some schools where there were a lot of students who didn't have a lot of resources. So I think it gives me some perspective. A number of different perspectives. Okay, so I wrote down those questions. Sorry about that. That was loud. Okay, let's keep going here. And what strategies and organizers do we want students to get used to and be able to implement while they're going through the year? Okay, let me check here. I'm just kind of making sure I'm not missing anybody who's wanting to enter. Okay, so, all right. So when I created these storylines, I really had in mind, creating these storylines for us, right? For biology teachers, for students of biology. And there's a number of things that I purposefully built into the units to try to really make them ones that are going to be easy for teachers to adopt. So this one I think is really important. The storylines, they live within one unit of the CED. Now, we'll get into that more in a little bit. So we kind of break down these main things, and then we kind of come back to them and give examples. Storylines, I think they're really motivating to students. I think they're motivating to teachers too. I've been so excited to hear from many of you during the year with messages like, I'm really enjoying teaching this class again, and I haven't felt like that for a while. And that's been just so invigorating and so exciting for me to hear. I love hearing those kinds of things. I want teachers to have that experience because I think that we have a job that can be very challenging, and when we're hopefully finding that exciting moment or that experience that really makes us feel like, yeah, this is why I'm doing this. I want teachers to have those experiences more and more, and I think that the storylines can really help deliver that on a pretty regular basis. I've also been working quite a lot on my website, which is being built for all of us. So it's being built for us as teachers to use, but also students too. And I'll show it to you a little bit later on, and we'll look at where it's at, and I'll give you some sneak peek on what's coming too. And okay, so teacher drive resources, I think a lot of you are familiar with that, but we'll kind of hit some highlights with that too. Just talk about what's there. And then just, another, I think, important part of the storylines is that there are, I think, very focused class activities, that the time in class is used really wisely. I think sometimes there may be this impression of storylines that it's very loosey-goosey, that the students are sort of creating their own, choose your own adventure book kind of class experience, and that you've got students going off in a lot of different directions, and each day is kind of unpredictable, and that's not these storylines. I teach in a pretty compressed schedule too, so I don't have the time for kind of wandering around through the curriculum. I feel like it needs to be very directed, but there's still a way to create that feeling of the students that they're kind of creating this class along with you. They are, in fact, but I think there's, it's very directed in how we kind of move them through the units. And you'll see that as we do some examples here, discuss some examples. Okay. All right. So this is one of the big points. This was really important to me when I got into designing these. Now, I will put it out there. Unit 1, it does cover still the chemistry of life. I threw in a little bit of natural selection into that first unit because I wanted there to be a context. I think that Unit 1, I mean, pardon my pun here, but I mean, Unit 1 is really dry, which is ironic, right? Because it's properties of water and the chemistry of life. But I think a lot of people would say this is a unit that is not their favorite one to teach. And so a lot of people out there, talk about, hey, I want to bump ecology and do ecology first, or I want to move evolution, natural selection, and start the year with that. And then circle back and do those, the chemistry of life. I didn't want to do that because those 2 units, 7 and 8, are the ones my students always say are their favorite, right? And here's what I was worried about. I thought if I put those units up front at the beginning of the year, then I'm wrapping up at the end with 5 and 6. Now 6 is probably, if you do 7 and 8 first, right? 6 is probably, a unit that I think I'm fascinated, but I think for a lot of students, 6 is a really hard unit. If that's your last unit, when maybe some of your students are starting to tail off on their motivation, I didn't want to do that, right? I wanted to keep those highly motivating units of ecology and natural selection at the end of the year, when getting students motivated can be tough. Like I said, I did throw a little natural selection into Unit 1 to give a context, to tie together properties of water and macromolecules, and it all fits, I think, the way that it came in. But the other storyline units do follow the CED units. So, Unit 5 covers heredity, and it's not going to bounce around and jump between 6 to 5 over to 7, then back to 1. I think there's some important reasons why I didn't want to put it together in that way. And I'll get into those as we go through this. So one of the, I guess, the really big reasons for this, you think about the way that you have already, if you've been teaching the class for a year or 5 or 10 or 20, you've put a lot of work into curating your classroom resources, your textbooks, a lot of things that you're using, right? And if a storyline is suddenly going to be jumping around between all these different units, then you have to shuffle everything, right? Here's this section of the book that we're going to read, and then we're going to jump over to this other section. Oh, Jennifer, personal progress check. The unit personal progress checks from AP Classroom. So I find that those are really helpful for students to use at the end of a unit. If you have a unit that is, a storyline unit that is distributed, parts of Unit 6 maybe, and parts of Unit 7, and parts of Unit 5, and parts of Unit 1, how do you use a personal progress check? I couldn't wrap my head around that. I thought about it. I couldn't figure out how to do it. And maybe there's people out there that have that idea about how to make that work. I didn't have an idea about how to make that work without doing a ton of extra work myself. So I thought it's better to have these storylines exist within a unit, and you can tell a story about heredity and do storylining. You can tell a story about cell communication. You can tell stories about energetics. You can tell stories within these units. So I figured, why make this more complicated than what it needs to be? I'll say more about that in a little bit. I think that the framing of this, to keep the storylines based on a single unit, I think it really helps to ensure that the lesson sequence is coherent, right? Here was my concern. If I was looking at doing sort of an integrated unit where it's multiple different units kind of incorporated all together, that's interesting, right? I think that might be really fascinating for teachers or people who already know all the biology, right? Because we're fascinated and we're seeing all these connections. Now, when you think about how Unit 6 is structured, and we're starting out with, structure of DNA and RNA, and then students are learning replication and then transcription and translation, learning about, regulating gene expression, like that's a very logical sequence. And if you were to take those different ideas and kind of pluck them out and drop them here or there throughout the year, that might make sense to the teachers, but is it going to make sense for our students? Like, is it going to be something that, and not just your top students, is it something that all of your students can really follow? And I worried that it wasn't going to be that, right? So I thought I want to stay within a unit. And then it gives the benefits of, within a textbook, you're kind of focused within, the chapter or a few chapters that might match up with that unit. We're able to use, personal progress checks within AP Classroom. We're able to use the unit resources of the daily videos within that unit. And we didn't have to recreate the wheel for a whole bunch of things, right, that are already there and they're working. So why not take advantage of them, how they're structured already? Now this one's really kind of looking at, some of the main features of storyline units. So storyline is going to be anchored by a phenomena. Now sometimes it's one, sometimes it's multiple. Kind of depends on the unit, right? So I typically will start with a brief video. Sometimes I start with a story. Sometimes I tell a story and then by the next year I've created the video version of that story. That's what happened with Unit 5. Students are going to generate, after they view this video, a driving question board. So they go through a process of, they're presented with something that is not easy to explain in the moment, right? It's something that you'd have to dig in. You'd really have to learn more about this topic to really fully explain it. And the students will individually generate a number of questions by themselves, and then they will work collaboratively with the team to generate the best questions that the group can form. And then we'll accumulate all of the best questions and create a driving question board for the class, which is going to be something that we use throughout the lessons. That same day that we do that phenomena with the video and the driving question board, I have students create an initial model. Now, this is not actually something I was doing when I first started the storylines, but then Lisa, she's in the meeting, she introduced me to this just last fall, and she said, "Hey, I'm doing this." And I looked at it, and I'm like, "This is incredible. I got to be doing this." So I added it in. So yeah, it really much appreciated that collaboration. I think that can happen within our community. So this is the student's first stab at trying to explain the phenomena, right? And they'll get bits and pieces of it. They might remember things from bio, and then they're going to learn more along the way to really be able to answer and explain what's going on. The left side processing, we'll see this when we talk about the notebook. I'll call them LSPs for short. And it also helps students remember label, scale, plot if they're thinking about graphing. But those are practice FRQs. And I think it's really been a big thing for me to fit those in because they support the storylines, but then it's constant practice writing for FRQs. So it's really, I think, a win-win with those. The main features too, so we're looking at readings. Students are doing daily videos. Students are working on LSPs. So they're doing that as homework. Now we're going to bring elements of that into our class discussions, especially as we start a class period. We may select certain questions from the LSP and talk about, hey, how would this question get scored if it was on the AP exam? And have students do some group discussion and reflection and some evaluation, peer evaluation, self-evaluation. We're doing labs, we're doing simulations, we're doing POGLs. The students are writing a lot in class, and it's all designed to have them build those science skills that are really important on the AP exam. Because we know that they're on the AP exam going to need to do transfer tasks. You know, we can't possibly cover every research paper that might get used by the College Board as they develop the next test, right? We can't possibly predict that. So we need to be giving students the skills to be able to read a text passage, to interpret a graph, you know, to be able to perform calculations. We want them to, you know, build all those skills. Finally, at the end of the unit, students will complete final models. And then they'll also, you know, I do give traditional assessments, you know, based on the materials from the College Board. So I'll put those together. I don't build them for the storyline specifically, because I think then it might give students a false impression that, you know, I want them to experience that transfer task, right? So if they do the storyline and then they take a test and the questions are related to those topics of chemistry of life, but it doesn't relate to the rock pocket mice or kangaroo rats, you know, I think that's kind of prepping students for what that experience will be like in May when they take the AP exam. So it's kind of the logic behind that. You're not going to hear the audio on this. So yeah, but this is the trailer on Unit 1. I'll just show you a brief part of it. We won't look at the whole thing. So this was initially inspired by my kid's pet hamster. It's a whole story, so we won't do the whole thing. But I was already using a lot of the HHMI rock pocket mice resources. I had developed over the years some labs that were very much aligned with the kind of concepts that are covered in the rock pocket mouse video. So it seemed like a really good fit, right? You know, and we get into all this idea of adaptive coloration and natural selection, but how do you bring in water, properties of water? How do you bring in macromolecules? So, you know, I wanted to think, okay, well, what are the other challenges that these animals are facing in the desert, right? Well, they're facing predators. They are, you know, and I don't tell the students a lot, right? I say water and students are going to think, well, there's not a lot of water in the desert, right? There's high temperatures in the desert. I want them kind of revisiting, thinking about what do they learn about chemicals and carbohydrates, lipids and proteins and genes back in biology when they took that in 9th or 10th grade, and just asking them to generate a whole bunch of questions, right? And they'll go through that process together and then generate a driving question board. So this is an example one. So these are questions coming in from the different groups. I'm not showing you that slide, but I'll put a QR code on my slide. Now, some of you may have local requirements that students can't use cell phones. So then, you know, through your learning management system, you put out a link to a Google form, but I have them, you know, enter the questions into a Google form, and then I can really quickly copy paste them into my slide presentation. So I've got my driving question board created like boom, really fast. I used it to post notes and posters too. Okay. So here we've got our driving question board, and now I think there's the danger that we think, oh, I'm limited to these questions, but we can sneak other questions in. Once the students have created it and it's coming from all these different groups, if there's something they really should have asked, I am going to add that in before the next lesson, right? Or I might sneak it in before you know, a lesson mid-unit where I said, oh man, I really wanted them to ask about this and nobody did, so I'm gonna like kind of sneak it onto the board. Yeah, so that's a driving question board. And I think that this is good because this gets students thinking, these are our questions that we're trying to answer. You know, this is what I was curious about. And I think that really helps to raise their motivation and their engagement. Okay, this is coming back to that idea of coherent lesson sequence? And I'm trying to answer some of these questions kind of as we go through, you know, some of these different slides, right? So that coherent lesson sequence, again, we want it to be a logical sequence for students. So in Unit 1, we're starting with that driving question board, which is the intro video, the student questions. The initial model is part of that, you know, and then we move into an activity called Mouse Hunter, where we're doing data analysis. Students are learning how to graph and use error bars. They're learning about natural selection. Now, not a full coverage of natural selection yet, enough so that we can analyze whether or not camouflage is giving a benefit for the survival of these rock pocket mice, but not the full coverage. That'll come later in Unit 7. But it allows that teaching of the statistics. So I used to do Unit 0 and I dropped that. So it's really now just we start with Unit 1. You know, so after this, the students are setting up a seed hydration experiment. I'll show you some pictures from that. And that's the connection of water and macromolecules in this unit. So they're setting up experiments, they're asking questions, you know, working with null hypothesis. And then kind of mid-unit, we get into properties of water, surface tension, and evaporative cooling. I have them do the water POGL. They're doing AP Classroom topic questions. You know, and then later in the unit, the seed hydration experiment is coming to completion. It takes about a week to run that experiment, and they're able to reflect on properties of carbohydrates and lipids, polar versus non-polar molecules. We cover hydrolysis and dehydration synthesis reactions along the way. They're doing data collection, analysis. They'll do a claim evidence reasoning poster potentially with us. I got a picture of that on a later slide. Now this is going to be a change for next year. So because of the rewrite and because they kind of did more changes with Unit 1, I'm really going to change Unit 1 to put in part 1 of an activity called the rock pocket dots, and really trying to drive home the idea of natural selection and the fur color pigmentation, thinking about proteins and the connection of genes. Now, this isn't going to be a full coverage of protein structure and all the information about genes. It's kind of introducing the idea of variation, genetic variation, and introducing the idea of gene expression, producing different phenotypes, and what that means for these mice that are, you know, facing the selective pressure of all the predators that live in their environment. You know, and then we've got, you know, again, probably assigning the Unit 1 personal progress check a few days out before their exam, just to kind of help them process and see like, hey, this is the level of questions I'm going to see on unit tests or AP exam. And then the assessment comes, and the assessment can be varied. So we might do formatives throughout. Final model can be something that's done individually, can be done with groups. I kind of prefer groups because otherwise it's a lot to look at. And it's a good discussion then between students. Serves kind of as a review while they're doing that final model poster. Spiderweb discussions, some units, those are better fit, some they're not. And then of course the traditional assessment is going to give us data where we kind of start to see where are the students at, like what are they doing well with, what things might we highlight for more growth and improvement. Okay, so that's the coherent lesson sequence. And yeah, let's talk about motivating students. You know, I think that one of the questions that came in was, you know, how do I minimize homework and how do I motivate students to do the work? You know, because that was one of the big things that drove the development of the storylines. And I'm going to take a look at this comment come in. I was really struggling with having students completing work. Like I had a ton of students that just were not completing their work. And it was a problem, you know, and it was a problem because there's so much to cover and there's just not enough time to cover it all. So I don't know how you can have students really be successful in AP Biology if they're not doing some homework. But I think we want to keep the homework manageable. Yeah, Natalie, I'm looking at your question. There's something coming up where I'm going to address kind of what you're talking about there. So I'll get to that. Okay. I'll give you a rain check. So I think, first of all, motivating students, right? So I think we need to do everything we can to maximize both intrinsic and extrinsic motivation in our students. So I want to do everything that I can, obviously within my I'm not paying them, but so budget concerns, but I want to do everything I can to motivate them, right? So I think those driving question boards really help to, spark that intrinsic motivation, that it's their questions, right? The coherent lesson sequence, I think the students can see the relevance of it. I think the storylines make the learning more meaningful for them. Like Unit 5 starts out with this story where it's this like family drama because a girl, who's taking high school biology class discovers in class that she's colorblind and didn't know it. And she takes the story home to her parents and explains to them how color vision deficiency is inherited, and it sparks this huge fight between the parents, right? But I don't explain the inheritance of color vision deficiency that first day, right? We kind of introduce, here's what it is, right? The student discovers she has it and that it's genetic, and she goes home and tells her parents and explains it, and it's this big fight. But then the unit is like unraveling that story of how did that lead to this big fight? How did that get, this former coworker of mine, how did that get him in so much hot water where the parents were trying to get him fired from his job? So, you create those stories. Oh, Natalie, it's fine. It's fine. I'll be recording, so I'll post it up later. So, we want to get the students really engaged in that learning process. Interactive notebooks, I think, are organizationally, they're so important. Like here's, this is a semester notebook. So I have students do a semester 1, semester 2 notebook. So this is 5 through 8. There's another notebook for 1 through 4. And so it's just kind of like all the work that they're doing is going into this notebook. And I'll show you the digital, but the students keep paper notebooks. And then really it should be one of the most important pieces that they can use to help them review for their AP exam when they are getting, into the weeks leading up to the test, right? Earn the curve. People have different thoughts on curving. Some people say you should, some people say you shouldn't. I was like, what can I do to, kind of put out that extrinsic motivation? So I do something to have students earn a curve, not just give it to them. They have to earn it. And that's a way that I found that's pretty effective in getting them to do their homework and keep up with their notebook. Final models, makeup work can be a challenge. We got students who are out, who are missing. I think the website's going to help with that. I think the fact that students kind of see this coherent lesson sequence, it's not like these units are a la carte menu. We're not going and we're picking from resources that are available somewhere. We're not picking a lesson for 1.1 and then we're like, okay, well, what do I do for 1.2 or what do I do for 1.3? So I'm going to go and pick another lesson, right? And it might be that this lesson that I picked and this lesson that I picked are not really super clear about how they connect. Now, as biology teachers, we know how those topics are connecting, but for the students, it might not be obvious. When we've got that good coherent storyline, they're going to see that progression. They're going to understand. They're going to know, hey, I need to be able to show, that I've mastered these topics, even if I was gone for a day. And there's going to be ways that they can go and, take advantage of resources that are available to them 24/7, through the website and through other resources that they can do that makeup work, right? There's still going to be challenges. There's still going to be the student that doesn't do it, right? But I think this can reduce the numbers that are there. That's the website, labs and simulation videos. There's more and more coming. So yeah. I've got this slide again later, but this is an example of like a formative assessment. So I'm having those students, I print the driving boards for them. I print the driving question boards for student groups, and then we use them on a pretty regular basis for individual and group reflection. So this is one from Unit 8 that this was something I started doing just really at the tail end of the year. But I felt like it was a really good way for me to kind of track where students were at the end of class periods, and for them to like kind of explore their questions within the unit, and then I have them add new questions as we go through the unit. You can see those written down at the bottom of that. And that was really, I think that was a really good addition for my class this year. That was something that really was helpful. I'm kind of bouncing around a little bit here. So this is back to the seed hydration lab. So this was how it gets tied together. You know, how do we tie together water and macromolecules, right? So the story goes back to my children's pet hamster. And among other things, we were noticing that when we would give him seeds, he wouldn't eat them all right away. He'd actually like go down into his burrow. Like we had the big setup where he could burrow down into his bedding, and he would bring all the seeds, you know, he'd put them in the pockets in his mouth, and he would bring them down into the burrow, and he'd bury those seeds for long extended periods of time. And my kids started asking, well, Dad, why is he not eating his seeds right away when we give him? Why does he always like store them away down in his burrow? So I started doing research and found out that these desert rodents, because they're, granivores and they're eating these seeds, they actually take the seeds from above ground where it's really dry. They bring them down to their burrows where it's more humid conditions, and the seeds are hygroscopic. They're like the little packets when you buy a new box of shoes. They are absorbing water vapor directly out of the air. So they can boost the water content of their foods. Kangaroo rats don't even drink water at all. Rock pocket mice, a little tiny bit maybe, but most of the water that they get is coming from their diet. And so when they find these seeds, they take them down to the burrows. The seeds are hydrating, so they gain, water mass. And then when they're eating them, they get more water. So as I was learning this about our pet hamster, it was right around the time that I went to NABT back in 2023. My brain's thinking, well, I wonder if it matters what kind of macromolecules are in the seeds, how much water they absorb. So this turned into this lab, and turns out, yeah, it matters. And it was the perfect connector of properties of water and macromolecules. And thank you, Lisa. Yeah, I was proud of it when I came up with the idea. I was amazed that it worked. I remember reaching out to my former DC, Angela. She left to take a position at our board of ed as one of our science specialists. I text her random things sometimes, and she's like, "What is Noel thinking about right now?" I remember texting her something about seeds and being hygroscopic, and she's like, "What are you even doing?" And I showed her later, and she's like, "Oh wow, okay." So yeah, the seeds will be set up so that we got some in dry conditions, like you see, just sitting out on the rack, and some are in this humidity chamber to simulate a burrow. And it's really good, produces really consistent data, really good for data analysis. It's in that biology context. So there's activities out there that'll teach graphing and standard error of the mean and, looking at error bars and whether or not they overlap. But some of them are not really in a biology context, and this one is, so I like having it in Unit 1 to teach the stats and to teach a little bit of natural selection and to teach properties of water and macromolecules. I think it really kind of ties it all together. Yeah, because it's all like my theme here is hiding the veggies. So we presented this idea at NSTA when we presented this spring. Hopefully we'll be doing some more presentations next school year. So the idea is like, if you're a parent, hiding the veggies in different foods that you're cooking up for your kids, right? Like, sneaking like mashed up broccoli into spaghetti sauce or something, right? So the idea is like we're hiding these little skills and things that are good for students into like interesting theming of storylines. So the lab has all kinds of things in there, like we have science practices, big ideas, learning objectives. The students are asking scientific questions, they're graphing, they're learning error bars. And it's all in this context of how do these little rodents survive in the harsh desert? We're doing statistical tests. They're learning properties of water, evaporation. They're learning hydrogen bonding, structure of biological macromolecules. They learn how to calculate percent change in mass in Unit 1. So when we come back and do unit 2, you know, whether it's potatoes or carrots, whatever you use, like that's easy calculation now for them. They've seen it before, right? So it's that consistency where these units are kind of looping, like one unit is going to kind of foreshadow things that come later in the storylines. So that's, I think, a big benefit of them. You know, we get into the hydrophobic versus hydrophilic, and that's great because it's really emphasized in this unit, and then you can really bring it back in unit 2 with phospholipid bilayer. So I think that there's some really strong pieces with this. Okay, this is an example of a CER poster, you know, that students created last fall, kind of looking at the results of their data. And I mean, you can see there in the image, we analyzed coriander, cumin, and pepitas or pumpkin seeds, and we can see that there's, you know, quite a big difference in the change in mass for the coriander seeds and the cumin seeds as compared to the pepitas. You know, the pepitas are very high lipid content. Coriander and cumin seeds are very high carbohydrate content. And, you know, it was really striking for the students. It was like, wow, I was, you know, not expecting it to be that different, but there it is, you know, and they see it with the data. And there's a great final model that I can't claim. It's one of Lisa's students. I put it into this slide presentation too. So we'll see that in a little bit. So interactive notebook. I create a digital one for the students so that if they're gone, that they know what's supposed to go where. Now I actually started giving my students up front. This is actually a lab page. So this would be like if you're opening up the notebook, I'll open up to a lab page here. So if we're opening up to a notebook, you know, we might have a lab put in here. In this case, it was the transformation lab. So I had the quick protocol for the students here, and then I print the actual lab as a booklet. And that way the students can get it in that one spot. It's all together, and then we don't run out of space in their notebook. It gets thick by the end of the semester, but we make it through the year with 2 notebooks. So that's kind of the trick on printing the labs. And then I'll just do the other side, like that protocol sheet. I'll just print it at 80%. And I just big paper chopper and trim them for my students. And I still have all my fingers after doing this for a 1.5 years. So I'm hopeful that I'm going to keep them all for my whole career. Hopefully I don't lose any fingers. Every once in a while you worry with those things they're pretty vicious. Here's another interactive notebook page. So this is actually one from unit 2. But you see we're going to pull back things from unit 1. So in this one, they're doing a calculation of surface area to volume ratio, and they're looking at rock pocket mouse there in the image, but you also see a moose. Now unit 8 really explores the wolf moose study of Isle Royale. So we're coming back and we're looping back around to some of these themes that have been kind of developed through the different storylines in the units. And I think that's like really motivating to students too. You know, sometimes I'll try to bury little Easter eggs within units, you know, like an early assignment, I'll sneak something into it and won't explain it. And then later in the unit, there'll be something that kind of like unlocks it. Oh my gosh, that's why it was this weird thing that we didn't understand back in the second lesson. You can be careful with those things, but there's a one in unit 5 that I thought was pretty good. So yeah. That's another one for a later time. We're going to run out of time here tonight. So let's try to look at a few more things here. Earn the curve. We talked about earn the curve. So this is how it works. Basically, you know, if a student is getting a score on a unit test, that would be equivalent to a 5. And I know these numbers can change from year to year, but this is numbers that work for me with my students. If they're earning a score that would get them a 5, if they've earned the curve, I'm going to give them a 95. Now, if they get 100%, I'm going to give them 100%, but that's not super common. But to earn the curve, they have to have done all the work. All their homework has to be done. The LSPs have to be done. The notebook has to be done. Like we're constantly kind of looking at their notebooks. I actually have them digitally turn in a lot of their work. So like the layout of, say, their, a topic page with the left side processing, they'll submit that through a Google form to me so that I can just really quick, I can sort by name and boom, 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 I can see, the students' 12 assignments that are turned in for the homework. For that unit, really easy. I know that they did it all. The other like classroom activities, I'm kind of checking during the class period as I'm visiting with student groups. So I kind of know if they're up on things, right? They're not violating the phone policy. We do have a no phone policy in my county now. My county also requires that we allow students redos of poor tests. That actually is more, I do corrections. But if they haven't done their work, I don't give them the curve. So if they score 70% and they haven't done the work, they get a C, and I'll show them the curve and say, look, if you had done your work, you would have had an A. Now that's rare that happens, but it does. I'll say it's rare because the vast majority of my students throughout the whole year were doing all of their work because they looked at this curve and they're like, oh my gosh, that is doable. I can do this and I'm going to do the work so that I have this curve available, right? We also in our county we're required to give students a 50% minimum if they've attempted a test. So if a student scores 20, I can't give them a 20. I have to give them a 50. So, that right there, I think can be kind of demotivating to students who are like, oh, I do nothing and I get a 50. So if I'm doing all this work and I get a 50 still, like they're not going to do the work. So that was, another kind of motivator for me within, my system that I'm working in, how to get them to do the work. As far as like, one of the teachers was asking about, working in a Title I school and having students that maybe aren't doing homework. I think students need to see the relevance of the homework and they need to see value in the homework. I have over half of my AP Biology class qualifies for their farms students, but they do their homework. Some of them come in we have a flex block every day for 30 minutes, and some of them will come in and do their homework during that window. So I think with the right ways to motivate them, we get students to do the homework, right? And it's a big part of what we're doing because the class time itself, is activities for the most part. Oh, here's that final model. This one's great. You guys got to see this. Homework, am I checking for completeness or correctness? I'm checking to make sure that they did it. Completeness. I don't have time to check for correctness on everything. Those left side processings, we will discuss key ones. They'll do peer feedback. We'll discuss them in class. They can always come to that flex period with me and ask about some of them if they feel like they were really stuck. But I don't grade those for correctness because I think it just encourages them to use the AI to try to write it. And I want them to try to write that on their own, So I feel like you've made the solid effort. You're coming into class, you're reflecting, you're sharing ideas, you're doing the feedback with peers. I want them to get, that homework grade. And homework is 10% of our grade in my school system. So it's not worth a lot of my time to really dig in and try to, check homework for correctness. I need to build structures where the students are encouraged to do that more and more so that I can free up my time for other things that I think are more beneficial to my students. Yeah, so this final model, this is one of Lisa's students. This is so great. And the pictures really kind of tell it all, but there's a line in here that I highlighted and it's so small, I can't read it. So we get the thought bubble. So the student wrote, the seeds that mice store are made mostly of hydrophilic carbs, which attract water, which I thought was perfect, and I mean, look at this drawing here. This is great, right? This is awesome. The student has really understood a lot of biology here. Plus we got lasers, right? I mean, any picture with lasers, I'm sold. So when I got this in an email back in the fall, I was like, this is the greatest thing ever. So I'm happy to share it with you all. Man, I've got more stuff, but I feel like we were trying to get our students to really engage and invest and be excited about what's coming next. Website stuff, man, if you're exploring that, all the topics are on there. I've updated for the new CED. I think we don't need a lesson for each topic. Some of it is we assign the homework, like 1.2, elements of life. We don't really need a full lesson on a topic like that, right? Other things that I'm working on for the website, I'm working on daily video style presentations, like 5 to 10 minute videos about the topics that are going to be in alignment with the CED, but also be in alignment with the storylines. So that's current ongoing project I've got happening. There's also extension activities. I've gotten into podcasts over the past several years. So there's a lot of really good ones I found. If you ever find a good one that you think is a fit, send it to me because I'll get it added in too. But the website, the structure, so if you go to the website, you just kind of click on it and it takes up the website. If you're curious to learn, like, hey, what is the connection of, introduction to natural selection in Unit 1, it's going to bring you to it. There's a reading. My plan for next year is to have the students do this reading more so than, like, a lot of my students don't read their textbooks. Even when I assign the reading, they do the daily videos. I'm like, if they get the content, they get the content. But what I would like them to do in the future is like read the website, use the textbook as a supplement, and sitting right here or right here is going to be like that daily video style. Like, here's topic 7.1, the parts of it anyway that are relevant for Unit 1. And here's a discussion of it. Here's how it ties to the storyline. So 7.1 is not full coverage. So this is a new CED, and these are the parts that fit with this first unit. The rest of 7.1 is full coverage in Unit 7. But this kind of pushes some of that evolution to the front of the school year and allows for kind of that theme of evolution to be carried throughout all the units. And I'm a big believer in that. I think that that's really important. So that's that. Let me go back over here. I feel like I'm rushing. Yeah, if you just search my name, there's a lot of Google Lens things from stuff I used to do on Socratic.org, and now it just goes to Google Lens, and I'm not super happy with Google about that, but so the topic pages, they're rewritten for the new CED. There's daily video mini style lessons coming. That's summer task. There's labs and demos and enrichment stuff that's on there. The teacher drive, the resources are there. And some of the things that you're going to find: teacher unit guides, planning calendar, interactive notebooks. So I forget who was asking this earlier. I think it was Natalie. Natalie had to leave, but and then I just went the wrong way. So within each teacher drive folder in the units, there's a written out teacher guide. I do need to go back in and edit these for the new CED because they changed some of the the lettering and numbering of the topics and standards. So I got to fix for that. But for the most part, these are just, they don't need to be fully rewritten. They just need to be edited to, adjust for those changes that came with the updated CED. So there's one for each unit, although I think 7. I got to write 7. Ran out of time for that. So it's here. It'll be on the computer soon. And then planning calendar. One of the questions that came in, a lot of people said, doesn't the storylining take more time? I think having these coherent lesson sequences is super important because it allows you to map out your year, right? It's not loosey-goosey where students are choosing this adventure and that adventure, and then you, like, it's a month later and you just finished Unit 1. We can't do that. We can't do that in AP Bio. We're never going to finish the course. And I personally, I don't want there to be a self-study unit at the end of the year. I want to cover all the units with my students. So I map out the full year like this. This is from my this past school year. And it laid out, August through May. This is what's happening. And I have to change it sometimes a little bit along the way. We have a snow day or something, and I'm teaching virtually instead of, at school. But it really forced me into being really selective about the activities. There were activities that I used in the past that I loved, that were fun, that students liked, but it's like, you know what, this doesn't fit with the time frame that we have. And if I put it in, there's something else that's maybe a better lesson for the students as far as their learning and understanding that is going to get missed. And then it's like, well, I could put it after the exam, but then it doesn't help the students when they need it, right? So mapping out like this, I think is really important because it really kind of forces me into like this. These are the days that I have for Unit 1, and I'm going to get it done in that time frame. Because if I'm pushing every unit an extra day, you know, I mean, you've all been there, right? We know if we're at the end of the year and we've pushed every unit just one more day, there's no time for Unit 8, or we're only covering Unit 7 for a couple of days, or whatever it is that we're doing. And I think that's not something I want to do. So the planning calendar, I'm working on updating for next year. This is interactive notebook. So those are built for the semesters, but you can see it's a table of contents, and I don't have students keep an interactive notebook. So this is posted for them through a learning management system, so they can click through it and they can see where everything's supposed to be and what they're supposed to have done. I give them up front that first day after we've done the driving question board, they're getting copies from me of the note handout, the Cornell note pages, and the left side processing FRQ pages. They're getting all that, right? And next year, the push is going to be your notebook is set up for the full unit, at least the homework portion, by the next day that we have class together. We have A day, B day alternating. So, you know, if they get all this from me on a Monday, they have until Wednesday to have everything like glued or taped into their notebook. And I think that's reasonable for AP students. We'll see. See how it goes with that. This is, yeah. So a day in a storyline class is, you know, kind of like what you might do if you weren't storylining, right? You're checking in, having students maybe write some practice FRQs. You might do a short presentation on difficult topics that students might have a hard time with. We're doing labs and modeling activities and simulations, and these are carefully like curated and selected and kind of laid out. And the trick is that you're kind of getting the students to think that they're creating the sequence, right? They're creating the questions and they're answering the questions. So it's authentic, but we have the structure set up in advance. We know where they're going to go, and we know how to move them in that direction so that we can fit the unit into the time frame that we have available to us. Yeah. This is another interactive notebook page. This is looping back to like thermal regulation. So this is from Unit 8 with Moose, and they can kind of tie back to that Unit 2 with the surface area to volume ratio. So this is, again, that driving question board, formative. So this is kind of like at the end of class that they would use this. And that was the final model again. I snuck that in twice. 45-minute, no double period. So, Vanita, you know what? I'm jealous because I think you actually have more time in that model. But then, so you would take the way that the lessons are written up in the teacher guide, and I need to add more notes this next year for that. But some of the lessons that I'm fitting into one day, you would just divide them over two, and you have more time, you know, because you're going to have, you know, on average, if you have 45-minute, you know, you've got, you know, 5 minutes more every 2 days than I have. So it's not tons different, but you got a little bit more. But then you got transitions, so it's probably the same, right? So you'd kind of subdivide. And I'm going to be adding in for next year some ideas about how to make that work with the traditional 45-50-minute class. Question coming in, and I think that's where we're moving to next. Like, what kind of questions do we have? So I'm just going to take some questions from you all now. Yeah, let me see here. Checking out the website now, how much of the material is on the shared drive? It's almost all in there. Every once in a while I create something and I've forgotten to move it over into the teacher shared drive. So most of it's there. And if you're ever teaching a unit and it's not there, you just kind of send me a, you know, an email or whatever, post something on the Facebook group. So yeah, yeah. That you've been doing FITSIT. Yeah, the HHMI, sometimes I have to select which parts of the HHMI activities, and I may be looking at some of those and editing down. Valerie, my website, if you Google search just Mr. Pauller, Weebly, you'll find it. But here, let me get a link. Or you got it. Okay. It's easy to find. I'm very searchable. So it's in development still, but I think I've made some good progress with it, and there's more work to be done over the summer. Well, thank you all for coming in. Are there other parts of questions that you've all wondered about that I haven't kind of spoke to before we sign off here? Maybe we'll do another minute or 2, 3, 4 if we need to finalize questions from people. Yeah, so the question is about the Cornell style notes. My students have a textbook. I found that when I was assigning just the reading out of the book and only out of the book, a lot of them weren't doing the homework. Now that was before I was doing the Earn the Curve. I started now moving more towards pushing the daily videos, which are pretty good. I think that they're reworking those. I do think that I'm working on some that are going to be really tailored for these storylines, but also teaching the CED too. So, it might be helpful, Question, since was unfamiliar with the phenomena, struggled to know how to guide through the units. I think that the website, the way that it's set up now is, and I was working a lot on that over just the past like 2 months. I think that that's going to be really helpful. And as I rewrite those unit guides too, I think that those are really key pieces. And, also we're looking at the possibility of setting up those maybe monthly kind of collaborative meetings to kind of talk about the units as they're coming, and like, here's how you frame this. Here's how here's how you set this up. We're going to set your students up for success, and you set yourself up as a teacher to be successful. So it's just, adding in those support pieces, I think is my goal. So yeah, hopefully you all find good stuff. And do I feel the website gives the students too much information? That's a great question. And I thought about that, right? If I have a student who's so motivated that they're going to read through everything that I posted, like in the first few days, they're probably that student that's going to get a 5 in May. And then they bring things to the discussion, and they may uncover, even layers within doing a POGL or doing a lab that are not on the website. So I used to worry more about that kind of thing, like students trying to work ahead. And I mean, I think there's reasons sometimes to not give away everything. But and there's some things like with Unit 5, with the culminating, like, how does it end? I'm probably going to make that video unlisted and not have it on the website until like I'm ready to share it, and just kind of share it in a different way, so that students can't just go and find the answer right away after the first day of what happened to this family that caused there to be the big conflict. But yeah, if a student is that motivated that they really want to dig in like that, I think it's good. Notebook, I prefer the composition notebooks. When I have students try to use spiral notebooks, like sometimes it's okay. Sometimes they really start to fall apart, especially if they have the perforated pages. So I was pretty fortunate where the other class I teach is in the STEM department, and STEM, for whatever reason, my county, I can't say this. I got notebooks for my students, and I got notebooks for all my students, right? And a lot of my students get their own notebooks because they have a certain color, a certain style that they want to get. But I have a stash of notebooks where if I have a student who doesn't have one, I give them a notebook. So I know everybody's situation is a little bit different with that, but it's worked so far. Yeah, 80%. 80% size, you know, and then I just trim two sides, and then it fits. So that's how I do those. And I was probably going to do another like explainer on just setting up interactive notebooks. It's just like a short video even too. Right. Okay. Is there anything else? Anything anybody's wondering, wanting to ask about? I think we're all getting tired. I'm tired. But if there's anything else, so we can hang out for another minute or two. Do I have an idea for when the updates to the narratives and units may be completed? Thanks, Amy. Let me see here. Yeah, thank you everybody for coming in. I got a question here. I'm going to scroll up and look at this. So if I miss your message of leaving, thank you for being here. Where did that one go? Tim, idea for when the updates to the narratives and units might be completed. So my goal is I want to work on unit 1 and try to get that one done before we would do the collaborative meeting for unit 1, which is I think going to happen on June 8th or June 9th. So I'm looking to try to get that one rewritten by then. And then the others would kind of gradually be done over the summer. Am I going to have all 8 done before the start of the school year? I'm not sure, because there's a lot of other pieces of this I'm looking at too. But my goal is that I have at least the first semester done. Units 5, 6, 7, and 8 are not changing a whole lot next year, so they're there and available. It's really just the numbering and the identification of the topics that's shifted a little bit. So I need to rework those, but those might be things that I'm working on in the fall for 5, 6, 7, 8, so that they're ready for spring semester. So that's my best. I'll continue to try to give updates on where I'm at with things. Vanita wanted to do interactive notebooks for years, didn't have patience. Once you make the transition and you really commit to doing it, there's a learning curve, but I think you get there. And I'm still learning a bit too, and I think I can share ideas about how to make it not as painful of a transition. So yeah, looking forward to doing that. Okay. Some of you are checking out the website, looking at things. Thank you. Yeah, we'd love to have you take a look at it. Answer keys. I have not posted answer keys. I'm a little worried that they escape and get posted online and students start looking for them. I really want there to be discussions between teachers and their students. I want the Facebook group to be a community where people kind of share ideas. And then it's not just me, that it's the whole cohort of us that can kind of talk and share ideas. I don't think it is too ambitious to do storylines for a first year AP teacher, because in many ways, like if I had been doing storylines, if I had these storylines when I first started teaching AP biology, I think I would have been a better AP biology teacher my first few years teaching AP biology than I was just trying to, because what I was doing was this a la carte. Like I was going to different online places and I was finding this lesson for one topic and a different lesson for another topic. And it was this disjointed, like the lessons taught the topics, right? But for my students, it was like, I don't see how this one connects with this one. And to be honest, it was hard sometimes to tell them, like, other than this covers the topic, like, how are they connected? And sometimes I couldn't tell them that they really were. So I don't really think it's too ambitious. And I think with a community that we can build, that there's going to be a lot of support for people who want to do this, even if they haven't been teaching for 5 years or 10 years or whatever. Well, thank you again, everybody, for coming in. I think I hopefully got to all of the questions. And Brooke, if you're still in, if you're watching this again later, I think the website with the descriptions on a lot of things is going to be really, really helpful too in making connections and understanding the tie-in of the left side processing to the homework to the storyline. I think it's really going to make sense and click. That's my hope. All right, everybody. Well, I'm gonna go ahead and sign off. Again, thank you so much, everyone, for checking in tonight, and really excited about a summer break, but I'm really excited too to really build this community and expand use of storylines in AP Bio next year. So thank you, everybody. All right, let me go ahead and sign off now. So take care. Enjoy your week with students, and I'll see you all in the near future. Okay. Bye, everybody.